This conference will now be recorded.
Hey, Pierre.
Right.
Getting ready?
Yeah, we are.
Cool. I mute myself, just anything, let me know.
Okay. I'm gonna unmute. Shanta, can you talk? Shanta?
Hi, yeah.
Yeah, very good. So I can hear you.
Okay.
Thank you for all the ones that are already joining. What I will do is that I'll mute everybody. I will keep Shanta myself, and when Mo is joined, we're gonna start the meeting. I think we still have a bit of time. So meanwhile, Shanta, you can see the screen, right?
Yes, I can.
Okay, good. So we can do the micro check, like 1 2 1 2 micro. It's okay. So we're gonna wait another 5-10 minutes to get everybody to join. I'd like to thank the one who joined us today. I also want to thank the people that are joining us from Europe. I know it's very early in the morning for you, so I hope we make your morning worth, and we'll give you a good idea to go to work today. If there is anything, just put it in the chat. What I will try to do is to mute everybody, okay? And then you can write the question in the chat. It's easier for us to see if we have duplicate question and track if we have answered them. And then if we need you to clarify the question, what I might ask you is to unmute yourself and clarify the question with us. So the idea here today is really to do a 30-minute an introduction on the challenge of manual cleaning and the part washer, give you a little case study and food for thought of how it happened in the industry compared to what is written in the regulation, and then have a 30-minute discussion Q&A based on a specific question you might have, or things you have encountered, or customer question, or QA question, or operator question that you would like to solve. We have about 30 people registered in this webinar. We have 17 that have joined at the moment. So we're going to leave it another 10 minutes, and then we'll start. And we have a few equipment supplier and a few industry people who make the medicine who have joined as well. So I think we have a good panel of the different expertise. Okay, it's time to start. So for all the people who joined us earlier, I will start the introduction, and if people join us later, they will jump into the core of the business. Yeah. So first of all, thanks again everybody to join this our first webinar, and I'm going to go through a few logistic slides very quickly. and then we will go into the core of the discussion. Yeah. So um, 3 point, no deviation. We are design, build, CQV organization. So we have with us 3 experts doing this webinar today. Maurice Bardin, which is director of CBE, which is also a process validation expert, very engaged with the ISP, a lot of field knowledge and realization of project. Shanta, who work with us, a CQV expert, and was engaged in the execution of the case study we're going to present today, and have drawn the learning of this case study which we're sharing. So thank you, Shanta, for preparing most of the slide. And then I'm Pierre Penings. I'm the owner, CEO of No Deviation. And we like to share our knowledge. So for the people who knows me, I'm also very much engaged within the ISP locally, and there is a lot of knowledge available there. And so what we try to do today is to give you a case study. Okay, so to give you what really happened in the field when we are in production, and a sense of the reality versus the guidance. So we're gonna have 2 parts in that webinar. So the first part will be on the case study, a bit of review of the regulatory guidance, and then the second part will be 30 minutes of Q&A, right? So what you can see here, depending on how you set up on the tablet or on your computer, you should have that little chat logo. So this is where we would like you to type all your questions that we will try to answer the best we can. And then we could eventually share some other ideas. So if someone have a question and you think you have the answer, feel free to raise your voice over there. So if you want to test the chat, you can do so. So the first part of this presentation will have a regulatory requirement on cleaning validation, a very high-level overview. Then we will look at what we call the critical quality attribute, and then the principle of cleaning, what we call the critical process parameter in this case. And then we will go to talking about the small item and the use of part washer and manual cleaning in sink. So if you look at the regulatory guidance, and I'm not going to dive too deep into it because all of those are freely available on the internet, so you can scan the QR code or go on it. So you have the ICHQ 7 that give you a series of requirements for the cleaning validation, and we have take the PICS as the reference here, but the PICS or the EU regulation are very similar because they are developed in parallel. And you can see that there is a series of requirements you need to follow when you do cleaning validation. Not going to read them to you. We're going to talk a little bit about them later, but we're also going to talk about the challenge that are not in the regulation. And we say that you can see here that where manual cleaning of equipment is performed, it is especially important that the effectiveness of the manual process should be confirmed at a justified frequency. So we're going to talk about what it is to justify frequency and how can you prove that effectiveness. One of the interesting guidance I found, and again you have the reference here, it's the FDA validation for cleaning process, a guide for the inspection. So this is, I think they ask a series of pertinent questions that are not only valid for the inspection part, but are also valid when you try to design your cleaning process or when you try to design your equipment to be clean and your whole cleaning master plan. Again, here, when we talk about manual cleaning, we say how viable are manual cleaning process from batch to batch and product to product. So those kind, you should ask yourself those questions. And in this guidance, they also say that is in general, if you apply the quality by design principle, doing testing to prove that your cleaning is effective is not sufficient. Yeah, what we need to be able in a science-based approach is to justify every step of the cleaning. What is the role of this step, and what is the intent, and then demonstrate that we have met the intent of that different step. So in order to demonstrate the intent, we need to know why we are putting those different steps in the cleaning process. So in no deviation, we are familiar with this high level, and we try to put everything in perspective of the patient. So everything we do normally is intend to create value for the patient in terms of safety and quality and drug delivery. So that patient need a product. This product is done into a process, and this process is done in facility, equipment, SOPs, utilities. So the patient is expecting a quality target product profile, and that product is defined by a series of critical quality attributes that if we meet, we are answering the patient need. And then this process is actually controlled to deliver those CQA by controlling a series of CPPs. Okay. And so those QTPP, CQA, CPPs is what you can find in the definition of the ICHQ8. And then what we put in terms of designing of facilities and equipment with the ISP, we define what are the critical design elements which allow us to be in control of the process, which therefore will answer the need of the drug development, meaning building the CQAs into our product. Now, when we talk about cleaning, we don't really call those as CQAs from a regulatory perspective, but we have kept the CQAs in bracket in our presentation to say this is the reason why we are doing certain steps. Okay, so I think it's very important that all the operator, the QA, the site director, the maintenance people, they need to know what to do, but they surely need to know why we are doing things. Yeah? And for me, the CQAs are the why we are doing things, what is really the intent. And once we know why we are doing things, we can go in continuous improvement. So Shanta is going to quickly review in this case with you what are the so-called CQA we were looking in our cleaning validation or in our cleaning process.
Pierre. So, as Pierre mentioned earlier, CQA is the physical, chemical, biological properties that should be in appropriate limit to ensure the quality of the desired product. So, in our case, the desired product is a cleaned small items. These items are reusable items. So, when it is used in a process, we need to clean them, make sure it's within the spec, within the limit, before we can use them to another batch of product. So what are the CQAs that we are looking into? We have to achieve an absence of microbiological residue, absence of product residue, and absence of cleaning agent residue. So when we say we need to have the items to be absent from microbiological and product residue, it is actually from the batch itself. So when we use the item in the batch, we will have these residues coming together into the washing area. So we need to remove them effectively and to use it for the next process. When we say it has to be absent from the cleaning agent residue, that is the cleaning agent that we use during the cleaning process itself. So we have to make sure it is removed so that it doesn't interfere with the next batch when we use the small item in the subsequent processes. And finally, we have to have the items to be visually dry and clean. So why do we need the items to be in a dry state? Because after we clean the items will be individually packed, usually individually packed before it is used for the next batch. So if we have water droplets in the items, it may cause the proliferation of microbes, which will contaminate the next batch and states. So this is the target that we want to achieve when we clean a small items. So if we go to the next slide, we will see how we want to achieve the target. So we achieve the target by manipulating or taking into consideration of this items. This is called a Sinner cycle because it was introduced by Sinner in 1960. This is actually a principle of cleaning. So it says that time, action, chemical, which is the cleaning agent that we use, and a suitable temperature will affect the CQA that we want to achieve. So these are the critical process parameters. These variabilities will have an impact to the CQA, and so this should be monitored and controlled to ensure that the process produces the desired quality. Changing any of these parameters will increase or decrease the effectiveness of the cleaning process. So during cleaning, when we want to validate a process or when we want to test out a process, we need to make sure that these parameters are within within the specs, so that we can achieve the target, which is to have a appropriate limit in the CQA. The next slide is about the roles of policies and procedures. Pierre will explain about that. Pierre?
Yeah. So again, you see here the CQAs and CPPs we were talking earlier. So in order to have this achievement, this is why we are cleaning. This is how we're going to control those parameters. I need to have process parameter, and I need to have critical design element that allow me to control those parameters. Now, the control strategy overall in the pharmaceutical plan is more complicated than simply controlling parameter. So basically, in general, you have material that come in, you do a process, and you have your equipment that are used by operator, which are in the facility, and from there you come out with a product. The material that come in and the product that goes out, and how you manage all your people and the flow and things like that, this is defined and written in your policies and procedure, right? So my quality testing is only valid if all those people here and all those facilities and things like that are behaving the same way as where they were behaving during the validation of my different process steps. And now you will understand that the people are a big variable in those. And typically the discipline, the behavior, and the training of those people, the training being trying to control the discipline and behavior, is what will allow me to remain in control. And the more people action I have in my facility, the more careful I need to be in the discipline and the behavior, or try to build systems that are mistake proof, that do not give the opportunity to the operator to make any mistake. And we're going to see how that is important when you go in manual cleaning. So 2 things you need to keep in mind, or 3 things, is what are the CQA, what do we try to achieve with the cleaning, what are the parameters that I can play with to achieve those, and who is manipulating those parameters, and how I need to define those parameters. So this is the high level for those which are not familiar. 2 pictures to illustrate what is a part washer and what is a manual cleaning in a sink. And so typically the part washer will come with an automated system and a series of a bit like your dishwasher or your washing machine at home. And then the sink is like making the dish in your kitchen. And so there is pros and cons of both, and Shanta is going to quickly review those pros and cons of the part washer and the manual cleaning.
Okay, so we have many pros in washer and some cons in manual cleaning. So as the name suggests, manual cleaning is a manual process where it is controlled by human, whereas washer is an automated process. We just have to validate the washer and at a certain frequencies, and we just have to put the items inside, and all the activities will happen by itself in the washer. So we will get a dried item from the washer. But in the manual cleaning, since it's a manual thing, so every step is controlled by humans. So human will, the trained personnel, of course, will perform the activity and until the final step. It is a manual cleaning is a labor-intensive process, because a person, actually 2 of them, want to perform, want to check, will have to be at the station at all times to perform the activity. Whereas in the washer, there is very less manpower required. Once we load the items, we can come back after a few hours depending on the time, and to retrieve the items and go to the next step, which is the packaging. However, if we want to clean complex item, it is most suitable to use manual cleaning to clean complex items. There might be some items which is not so suitable to be cleaned in washer, especially those which has grooves and very complex. In a washer, when a washing is performed, there is an audit trail. We can always get the printout on what happened during the process, and we can attach it in the record for further references. But in a manual cleaning, there is no audit trail. We will have a checklist, but it will have a variability from an operator to operator and product to product. So since there is no audit trail, there's only cleaning checklist. So the credibility and variability can be challenged during any inspection or audits. But manual cleaning can be a very good backup for washer, because in washer we are dependent on a machine. So if there's a breakdown in the machine, we may not be able to perform the cleaning activity when we need it during the process. So manual cleaning can serve as a backup for washer. So you might think that there are a lot of advantages in washer. So why don't we just like dump manual cleaning, don't use manual cleaning, and just wash everything in the washer, right? So if you go to the next slide, this is the ideal system, ideal photograph that we take from the washer. So we can see that we just have to load everything inside, and it will just do the process and give us the cleaned item, right? But if you see the items that we are going to load to the next slide, the items that we are loading and the items that we use is this. It's not like one or two items that like we use in the at home. So it's a lot of complicated items. We'll have tubes, reusable tubes, valves, and at the same time we will have some beakers and everything. It depends on the process. So it's very complicated to put everything in the washer. So in some process where it requires, it uses a lot of these small items, using only washer might not be so ideal. So we might have to have a manual cleaning in sinks in process. So the project that I was working on, if you go to the next slide. So you can see that, okay, so when I joined the project, there were around 40 items, type of items, which were cleaned, small items. So all those 40 items can be cleaned in manual sink, but only around 24 of them is validated and tried to be washed in a washer. So what happens here is, if the washer is not available due to any reason, or maybe there are not enough items to be loaded in the washer. So what we did, like we just clean everything in the manual in the sink, and we proceed with the process. The intent to have it is so that the manual wash will be a full backup of the washer. However, during an inspection, there was a challenge that there is an inconsistent operation. So does that mean if you can put a valve in a washer, and at any point of time, the next day or the day after, you can even clean it in the manual cleaning? So the rationale of putting an item in the manual washer and washer was not accepted during the inspection. So what we did, we did this project that in an ideal stage, go to the next slide. In the ideal stage, we want all items from the manual cleaning to everything to be shifted into the washer. So we have a remaining 16 item type of items, right? So the target was to validate everything in the washer. So how we want to do it is what we will discuss in subsequent steps. But before that, if we go to the next slide, I will explain to you about the purpose of each cleaning step. This is common for both washer and the manual cleaning. So just imagine it as the dishes that we do at home. So we will first do a pre-rinse. So we'll have a soiled item. We will just use water to rinse the items to remove all any soluble and non-adhering residues. Next is the main cleaning step, is the which is the cleaning using the cleaning agent, which in our case is sodium hydroxide. It removes all the dried residues and solubilized soil by degradation, heat and wetting with cleaning agent. Because the items that we clean does not necessarily clean immediately. There is some acceptable hold period, which we call the dirty hold time. So when there might be some residues which has dried, so we will clean this using an appropriate concentration of cleaning agent. Then after we clean using the cleaning agent, the next step is the intermediate rinse, where we will remove all the soluble and non-adhering residues, and also to remove the suspended solubilized soil and the cleaning solution. So when we go to the third step, the job of the cleaning agent is done. So now we have to remove the cleaning agent through some intermediate rinses. And finally, to ensure that we have removed all the remaining residues, we will do only one final rinse and put it for drying and then packaging. So if you go to the next step, I've showed you the steps and parameters that is used in the washer and the manual cleaning in sinks. If you can see, the steps are the same. The parameters, which is the CPP that you can see on your top right, the Sinus cycle, the time, the action, the chemical, the temperature, the one that is different, we have highlighted in red. So if you can see, we will soil it with the soil and put it for the dirty hold time for some hours, and then do the pre-rinse. So the difference here is that in washer, there's a pressure of water which will circulate through the washer, but in manual cleaning, we will recirculate the water without a significant pressure. So the time differs, but it is very negligible because it's very minor changes in the time. Next, it's the cleaning using cleaning agent. So the temperature that we use is 85 degrees Celsius. So when we as you can see, the parameter, the temperature is one of the critical parameter, right? So in high temperature, the efficiency of removing the solubilized soil is high. Next is the intermediate rinses. So we will perform a few intermediate rinses to ensure that we have removed all the soils and cleaning agents. And finally, we will final rinse and put it for drying. So there are a little bit difference in the action that we use, but other than that, the temperature and the chemical is the same between washer and manual cleaning. So
Just
yeah,
just normally, if you reduce the action, you need to increase the time, and then it's in the Sinner cycle, you can see that. And so in manual washing, what people do most of the time for some of those items in order to gain consistency is to, as much as possible, soak the item in the sink. Yeah? And soaking the item in the sink make very little action because I don't have any diffusion or forced diffusion. So I'm just counting on the normal concentration gradient that will happen between my cleaning solution and the surface, whether in a part washer due to the injector and the falling film and things like that, I will create a much more turbulent flow, which then make the cleaning more efficient because I increase the action I have and therefore I can reduce a little bit the time. So that's why you can see that there is some difference in the parameter I'm using. Also in a washer and in a manual cleaning, here I'm using a recirculation of water, whether here I have a pump that is recirculating. So the intent is the same, is recirculate the water, but the critical design element in a washer compared to manual washing are slightly different.
Yes. Okay. So we'll proceed to the next slide. So we have seen this slide before, right? So our target is to have 0 items to be cleaned in the sink and all the items to be validated in the washer. So what we did to achieve this target, if you go to the next slide, you can see we performed some steps, some assessments. So all the small items which were used in the production, be it in the buffer prep area, fermentation, or downstream area, all the items were assessed for the feasibility to be cleaned in washer based on complexity and number of items. So what we did was we took a list of the items and how many of the items were used in a day in a process area, and see if they can be cleaned in a washer. So what we did, we modified the washer injected trays to accommodate all the small items. So as much as possible, we see whether we can modify all the trays to accommodate the items so that the items can be cleaned effectively. And we found that there were some items which were not feasible to be cleaned in the sink, in the washer, due to the complexity of the item itself. And those items were the one that we validate in the manual cleaning, and the remaining items we put it in the washer and do a new validation to validate both the manual cleaning and the washer. So in the assessment, if we go further to the next slide, these are the examples of items that we have assessed whether it can be cleaned in the washer. So if you can see, we have a lot of stuff like flow meter body, pressure gauge, which is not here, connections, diaphragms, clamps, and scissors. So we did this assessment to see if we can modify the injected tray model to clean these items. And then after we do all this assessment, we went to the acceptance mode, which in the next slide you can see there are some items which are very complex, which cannot be cleaned in the washer with the modified tray, injected tray. So these are some items that you can see in the photo. This is a sampling valve, which is very small, 0.5 inch. So it's very complicated to modify a tray to clean this. So the ideal situation, the realistic situation is to clean this in the sink. So if you can see that on your right is the transfer piece, top right. So it's very small. The diameter is very small to be able to fit in the washer injected tray. So there are some examples that I have listed out here. And also pressure gauge, which is not in the list. They are sensitive materials which cannot be cleaned in the washer. So we have to clean it in the manual cleaning. So what we did next was to determine the maximum number of items to be cleaned in a day based on the process requirement. And then we validate the manual cleaning and the washer. So if you go to the next slide, next step is to validate the items. So the routine cleaning, when we want to validate a routine cleaning, so we have to change some of the parameters to make sure that the routine is able to clean properly. I will repeat that to you. For example, validation has a maximum number of items used. In routine, we might not have all 10 sampling valves to be used on the same day. But during validation, we used all 10 sampling valves so that we will have a maximum number of items. And then all product contact items were soiled in the worst case soil. During routine, not all items is being used in the worst case soil area. There are some items which is used in buffer, which might not be the worst case area. So, but in validation, to create a worst case scenario, we have soiled all the product contact items with the worst case soil. We also validated with a reduced contact time during pre-rinse and cleaning with NaOH solution, sodium hydroxide solution. And finally, in the manual cleaning, we have one less intermediate rinsing. So if we can prove during the validation that at this situation, at this scenario, the routine, the cleaning is able to be achieved. In routine, when we do more than this, the cleaning will definitely be achieved. So if you remember the CQAs, right? Absence of microbiological residue, product cleaning agent residue, and visually clean and dry state. How do we want to validate this, that we have achieved the CQAs that we want? So that there are some tests that we conduct. So we will take sample from the final rinse and we will conduct the following test, which is the conductivity test. So the acceptance criteria is that it should pass the water for injection specification to prove that we have removed all the cleaning agent that has come in contact with the small items. The next one is the TOC, microbial content and endotoxin content. This is present in the product itself. So when it is within the acceptance criteria, we can use these items for the next batch. And finally, we will have to visually check whether the items were in clean and dry state, so that this is a visual inspection, so it requires proper training and everything. So when we can see that it is clean and it is dry, we will pack these items and keep it for the next storage. And Pierre, do you have anything to add?
Oh, so many things. I could talk for hours. But,
Yeah.
so basically, this is the intent of those presentation is to show you what you need to think. And in this case, the case study is based on an operating manufacturing that need to go back to the drawing board to rechange their cleaning procedure and their cleaning process to bring that consistency. I think there is two key learning here in terms of manual washing and part washer. The first one is if you look at those items here. When it's very difficult for when you are in the design of the equipment to think of all the items you will need to wash in a part washer here. So it's a little bit like, especially when you are in a green field and if you have a new project, and whether you are the engineer who need to design the part washer or the vendor who need to design the part washer, all you want is a list of items and have physically all those items. Sometimes the process is not completely designed and there's a series of items that the user doesn't really know. So I compare that to trying to define the load that's going to go in your dishwasher for a wedding banquet 6 months before the wedding, right? So you're going to have a series of cutlery and a series of plate and cup, but exactly which plate and cup and which cutleries and things like that, it will really depend of the meal we're going to choose and how many guests we're going to have and things like that. When we are asking the same to our user, when we ask him, can you give me all the pieces you need to put in the part washer so that 6 months before I can define those part washer. So there is an iterative process in designing a rack and validating your process that will happen during the project. What we see here is that in terms of design of the small item, we should minimize the number of items that goes in manual cleaning. And we should really try to say manual cleaning for me is the last resort. Everything should go in the part washer, and deciding that the manual cleaning is a backup of my part washer is not a good strategy because it leaves the door open that something is wrong with the part washer. I decide to bring it into the manual cleaning, which is always available. But then I have a lot of variability because in one batch, some item goes in the part washer, where in the batch after, they might go in the manual cleaning. And I bring all those variations in my manufacturing process where they're actually not needed. If the part washer is properly designed and maintained, there is no reason to have a manual backup, right? Then when you design all those items, this is really when we go from an R&D lab to a manufacturing plant without thinking of the manufacturability of my process or the scaling up of my process. Yeah, all those items that are used in the lab are okay to be in the lab because we're not in a full GMP environment when we do the process design. But when we are in manufacturing, we need high level of consistency. So we should think of how can I have all those designed better to suit the cleaning process, and the cleanability of your item should enter in the design of the different equipment piece and things like that. So so for us, this is the key learning, right? You might need some manual washing, but the manual washing, in my view, should be kept as little as minimum as possible. So I don't know if you have specific question. I'm gonna check in our chat. Okay, how is the recirculation done in the manual washing?
I can explain that. So in the manual washing, it is performed in the sink, right? So after we fill the water, what we do, we will connect a peristaltic pump using the silicone tubing. So we'll insert the silicone tubing inlet and outlet. through the peristaltic pump, and we switch on the pump. That's how it's done.
Okay. And so you can so basically you soak everything in your sink. You put the inlet of the pump and the outlet of the pump in a different location in your thing. You turn the pump and you recirculate the water. And you can understand that the action of a recirculating water is really like um, a laminar flow, whether the action of a recirculating in a part washer with injector and a certain pressure is really like a turbulent flow. So the action in a part washer is much better than in a manual wash. But the beauty of soaking everything is that all those little tubes would be completely soaked with water. Whether if I put an injector in it, I have no idea whether the injector will just shoot through or if I'm going to clean the internal part of those little tubing. Yeah, so that's why we sometimes still go to the manual washing. Hope that answered to your question. How to establish the validation strategy, the contact time, the intermediate rinsing time, and what are the different steps? So the question is, here we know the intent of those different steps, which will be the first, how many steps do I need to have, and do I need a pre-rinse and things like that. And the second thing is that how do I define all those parameters? So maybe Maurice, this is a question you can give a glimpse out. I know you can do a whole seminar on it, but how do you define those cleaning parameters? Is something you can explain a little bit? Did I mute Maurice?
Everybody. So you're asking me to talk to the how do I establish the validation strategy, especially on contact time?
Yeah.
Yeah. So it's iterative. Is the bottom line. So in a cleaning validation, we obviously don't set up to try and commission the cleaning as soon as we start, or validate the cleaning as soon as we start. So you would have done some trials with parts or certain parts that you think are representative. So you have an idea of the cycle, and you're looking to simulate that. And usually, in a real sense, when we present this, sometimes in a seminar, we might show you a beaker that we've been rinsing, or a coupon that we've been rinsing. But in reality, in a big factory, you've rinsed and tested a number of components. So you have not only the component data, but you also have that you can compare the various parts, and you start to get a feel for how significant some of these parameters are compared to one another. So, for instance, you get a sense of how significant temperature might be compared to contact time, and you can then use that to leverage some of your other cleaning. Is that a good enough answer? Does that explain?
Hi, Maris, this is Shanshan. Actually, I posted the question. I was more curious on like this case study we showed, we reduced the one intermediate rinsing step, right? I've seen some of the validation will probably reduce the rinsing time for the intermediate like 70–80% each cycle. So to decide that, do we need to use some stats to establish a study, or it's based on experience stuff?
I don't think it's either, to be honest, because what
Right.
tends to happen is it's not exactly like a manufacturing process where you simulate your way into the process and you understand what's happening with these variables. What happens is you end up in a sweet spot. You effectively do a trial and you end up in a sweet spot. And you've either got to have experience or data that helps you move around from that sweet spot. So it's not luck, because you land on it by doing trials, but really it's very few companies in my experience, unless they're doing repeat validation, cleaning validation, that actually know where to go to start with. They tend to arrive there more by luck or by precedent. So so it's difficult sometimes to sort of try and apply absolute science to the parameters. I think you're better off to sort of think, what would happen if I move by 10 or 20%, and how does that affect? So that's really the strategy that I would be doing, and that would be by experience. So for instance, you talked about doing temperature at 85 degrees C. What if it wasn't 85 degrees C? Well, clearly cold's not good enough, but what could be appropriate? So you might do 65 to 85. It might be the strategy that you do, and it helps you understand the timing so if you're using a manual cleaning and you're not replacing the water but you're recirculating it, it's going to cool, right? So it is a bit of common sense, I guess, and pragmatism. And so what we're trying to do with cleaning procedures quite often is land at that sweet spot or that target all the time. It's not so much about operating around the target.
Okay.
Cool.
So
Thank you.
I think what is important to keep in mind is those steps, yeah, is to why we are doing the different pre-rinse and things like that. And I think if we know the why, and there is a bit of science based on your product, yeah. So if you do for example, a saline solution, and that's all you have, well, you don't need to put a cleaning agent, because a cleaning agent might be a higher risk than simply rinsing with water. Yeah, if you work with protein, yeah, definitely the pre-rinse, and you need protein cook. So you need to do a pre-rinse cold, if possible, not to cook. So if you work in a blood factory, a cold pre-rinse is a really good thing to do. Otherwise, you're just going to coagulate all your agents and things like that. So those are the items. Sometimes you might need to throw an ultrasonic bath. We were working in a facility that were using eggs to make the product. The only way to remove all those albumin and things like that was the ultrasonic bath for those little parts. So I think it's important to define why we have the different steps. Then we will have a series of parameters, like Maurice have said. We work with Coupon to define the cleaning strategy, so see what is working, what is not working, and what have a big effect on the cleaning and what have almost no effect. And then you go in your cleaning validation program and you demonstrate you can achieve that, and you don't really need to test it to the limit. What you need to have is a consistency. We also have a question: is how do you create the worst case soil model for the validation? I think it's a very interesting question, and it's sometimes a very good challenge here. So I don't know if Shanta, you want to answer this one, or we bounce it back to Maurice.
We can bounce it to Maurice.
Okay. So Maurice, how do you make the assessment to say, okay, this is my worst case I'm going to use? Because everybody say you need to use your worst case when you do your dirty all time. What you're going to do is that you're going to spray your solution on it, and then you're going to do your cleaning validation with the worst case. So how do you do that worst case solution?
My experience is that it's usually an overkill situation. So again, it's not about precision. It's about creating a situation that is considerably worse. So what happens is, in a typical cleaning validation, when we break it down, there are a lot of assumptions going on. We've got assumptions around recoveries. We've got assumptions around areas, and often in situations. And so the worst case soil residue is also subject to assumption. So in my experience, we're better off just to create something that is significantly worse. And even that could even be representatively worse. So what I mean by that is perhaps even using a surrogate and showing that you can clean it. And we do that quite often because we group. So sometimes and it depends on the type of plant also, but in multi-product plants we group, which is a way of selecting the worst case. In a large monoclonal antibody plant, the worst case is not so much the grouping of the product, but probably the worst situation, which might be concentration or solids content or something like that. So they tend to be physical situations that you're looking at, and you do need, I think, experience. And the sort of experience I'm talking about there is what's gone wrong. So for instance, in cleaning something like a freeze dryer, the worst case might not actually be normal use. It might be abnormal use. So if you lose the vacuum on a freeze dryer and you boil the product that you're trying to freeze dry, that's actually the worst case to clean, potentially. And so it's difficult, I think, to give a straight answer, but apart from the fact that it should be an overkill.
What we do also generally is to try to identify which piece goes on which part of the process. Yeah. And then you could do a worst case for upstream and a worst case for downstream, and lighter your validation over there. Or if you want to put everything in the worst case solution, and then you do upstream and downstream in one go. What you could do as well is you need to have the list of all the chemical and material you're going to use in your facility and that are going to be in product contact, and make sure that in all of those you can, for example, highlight the one that is less soluble, that the one that is the highest viscosity, that, and then start to say, okay, this is all the worst case things that are going to get in contact, and this item is worst case than this item for this and this reason, and therefore that's the one I'm going to use in my worst case solution for my validation. So there is a good understanding not only of the process you're going to do, but all your manufacturing steps that need to be established before you can define what is the worst case validation solution. Yeah. And if you didn't think that through, it's a lot of data mining.
The other way I've seen that's quite practical, I think, in terms of a risk case, is thinking about that Signer model, but thinking about it in reverse. So if you're thinking about a particular cleaning situation, then if you think about the concentration of the soil and the effectiveness of the temperature, right, and the action that you're applying, because really worst cases in a lot of instances more to do with cleanability than anything else. I mean, if you understand the toxicology, if you want all the risk, that isn't really going to change much through the process. It really comes down to the amount of product that's left behind. And so that's really a cleaning issue. So if you were to use this circle backwards, so, you know, the situation in my process where I see the least action. So in other words, if it's a CIP process, it's where the velocity is at the least, right? Where I'm less likely to see the chemical. In this case, I'd be thinking more about the amount, the concentration of the soil. Where's the temperature most likely to not be where I expect it to be, and that can help you decide the worst case, and then you can use that to model the cleaning parameters that you would require. So you can take that offline into the lab, simulate that situation, and see how different that is to what you consider to be your normal cleaning. And you could use something like time. You could say it takes me 5 minutes longer to clean that to the same level as it takes me to clean the normal. So that gives you a sense. You're dealing with you're dealing in sort of understanding of orders of scale.
Okay. Tony, thank you for your comment, Tony. So Tony is from Factory Talk and is also one of the star players of ISP. He's using a system called Tulip to manage the manual operation. So I agree with that. Today, when we say there is no audit trail for the manual operation with the Industry 4.0 and all the different tools and paperless solution and MES solution we are having, it becomes more and more easy to record the different steps of what we are doing. And you can see also in our manual cleaning, I have two questions asking me, but how do you wash manually at 85 degrees? I'll give you a Belgian answer on this one. It's the same way as you cook French fries at 180 degrees. Okay, you don't put your hands in it. So basically you have a basket, and that's why we soak the element with a recirculation. Then I can take out the basket, drain the water, and rinse with cold water. So in this case, when you see all those small items, it's not even feasible to go and scrub all those little inner tube and things like that. So we really soak the stuff with recirculation. And as you can see, I'll try to get my pointer back. As you can see, when we soak those things, the real challenge is that I've almost no mechanical action. Yeah, I'm really depending on the chemical time and the temperature, and the only action I have is the diffusion of my chemicals and my soil getting dissolved and things like that. So in those little items, what we do is that we soak them. So we never ask our or operator to put their hands in an 85-degree water, and if they have to do is mostly to pick up a basket, and they have thick thermal gloves and things like that. So again, manual cleaning for us is not the best solution. Another challenge you can have with manual cleaning compared to a part washer is that a part washer, it's very easy to segregate the clean side and the dirty side, right? You load from the dirty side and you unload from the clean side. Now, when you are in a sink, the flow of clean and dirty is much more left to the operator behavior than being an engineering control. So what we are saying is that as much as possible, avoid manual cleaning. If it's not possible, think it through. And the way to think it through is to take all those elements and try to see, can I change my critical design element of my process? Can I change my equipment? If really I cannot, and that's where the end of the assessment and the difference between the ideal state and the current state is that while some of those bits and parts just need to be soaked and find a way to clean them. So this is how we wash things at 85 degrees, is that we soak them at 85 degrees. Another question is that the cost of the automatic washing versus the manual washing. Yeah, I give you that. There is a capital investment at putting a washer that is not the same as putting a sink, right? I think it's obvious. Now, I think there is a clear benefit of having a washer in terms of state of control that we try to achieve. Yeah. And again, Maurice have said we use in coupon, we also need to use a different material because there are different challenges. So if I take those equipment, plastic like this doesn't have the same adherence of the product as stainless steel. But what you will see also in a part washer is that the drying of the plastic take forever, and then you end up having drying cycle that take forever, because when we dry in a washer, we generally don't have the same mechanical effect that when I take compressor to dry the pieces and things like that. So there is a lot of things to what we say, it's beginning with the end in mind when you develop your process. And if any of those pieces is not properly washed and you start to have issue in your process, that's where you start to regret not to have invested in an automatic washer. Yeah. Now, what is really costing the automatic washer is the design of the rack. Because again, if you try to make those rack an iterative process, so we can see here in the presentation that at some step we have decided that to modify the injector tray and to modify the rack to put the new item. The level of effort to do that is big, because you need to do a new IQ for the rack, you need to do a new coverage test for the rack, you need to do a new validation for the pieces that are on those rack and things like that. So think it through before saying that we're going to put everything on those injectors, and think of the flexibility and the agility you want to give with your machine, and also define the right time to design and build the rack.
Can I say something here about that comment? So to me, a lot of people lose sight of what we do when we do a cleaning validation. So if you think about, I mentioned before the formula that we use typically when we do it. So when we're going to do these parts washes, these parts are generally part of a larger system, right? And that limit that's been established has usually been established as part of the larger system. And so one of the big considerations about the two choices to me is the location in the process. It may seem obvious, but if you think about the way the formulas work, when we use swabbing, and if we're using swabbing to establish a limit, we're making some assumptions based on the shared surface area about what can actually happen. And the further away we are from the filling point, the less critical that assumption is, in my opinion. And likewise, when we do rinsing and we think about shared surface area, we've also got an assumption, that's possibly a not as worst case. And so to me, the decision about a parts washer, if you're looking at that, becomes more critical as you get closer to the patient.
Okay. Thank you. Yeah, so there is a comment there on the worst case soil. The worst case soil need to consider the material with the least acceptable residual carryover. So for me, like Maurice have said, we do an overkill, and if really that overkill need to be challenged, we can challenge it. And when you do the worst case solution, you also need to think of the different material on which you're going to apply this worst case solution. Yeah, it doesn't stick the same on stainless steel or silicone or other element. We have answered most of the question, and I think we're coming to an end. So I don't know if any of Shanta or Maurice, you want to put an additional comment or a point we forget to cover?
No.
Good.
I'm good. Yeah.
Which grade of clean room classifier area is the manual cleaning performed? It's a good question.
Mm.
So for me, again, it depends what you do with the piece after. Yeah, if after that the piece is clean and you put it in an autoclave and you know it's cleaning an autoclave. If you just clean it and you don't need to sterilize it, generally, for me, I would say a grade C area, but that should be part of your control strategy. But a clean room generally will be a grade C area. Am I correct, Maurice? Or?
That's what I would have said as well. I would have just said it depends on the process.
It depends, but the worst case would be a grade C. You don't do washing in a grade B or a grade A, so that's clear. So for me, and generally, when you are out of your part washer and you have an endotoxin concern because you are doing an aseptic process, what you want to do is to unload your part washer under a laminar airflow so that you control the environment before you wrap it, and then you can go in your autoclave, and then you have a sterile product in the wrapping. Yeah? It's the same reason why we want to keep those clean in this case and dry is because we're going to store those clean equipment into a shelf. And if I have any concern of endotoxin and it's not clean, I have actually a good media for the bacteria to grow up and redevelop that endotoxin I don't want to have. Now, if I'm going to wash it and use it, well, eventually they could still be with WFI. If my next step is to do a WFI rinse or on equipment. So again, you need to understand why they need to be dry so that you don't make that statement and you don't over dry things for a long hour, where the first thing you're going to do is to put them in a buffer preparation with WFI and things like that. Yeah? So you need to really to understand why we have those different steps based on the process and the control strategy. That's how you need to work, and don't take them in isolation.
I think one of the interesting things that I think about for this is that if you think about water compared to clean hold times, that sounds really weird, but most people know that clean hold times, they can go for days, right? They can be days, 7 days, 3 days, 4 days, quite easily, but you can't leave water sitting. So I think people who work in a non-sterile environment, they sort of understand, or even if it's sort of the beginning of a sterile process, effectively if there's water around, then I think that's much more of a risk than the environment itself. So to me, I would be concerned about drying first before I would worry about the room classification, because I think, within reason, I think that water hanging around has more to do with problems than the room classification would do.
Yeah. So to answer our last question is, cleaning is not sterilization. Yeah, it's two different things. What we can do in cleaning is to reduce the bioburden, which then, NaOH or cleaning agent can have a burden reduction effect that will then ease the sterilization process, which is a log6 reduction. So the more I control, the better I am to go into my equipment. And then again, if I sterilize, and I take that example, if I sterilize a dirty frying pan, which I just cook bacon, I will have a sterile frying pan, but I still have a risk of pyrogen component in my pan that could eventually endanger the patient. So in my view, you clean first and then you sterilize, and the cleaning is to remove everything so that you have clean equipment and you can have an apyrogen or pyrogen-free sterile equipment. So the step is then, that's why we put the laminar airflow when we work in aseptic for wrapping the equipment, is to make sure that we don't put back pyrogen on the piece we just washed. Okay, that makes sense. Good. Well, it's about time, so I'll thank you all. What I will do is to send you all the a feedback form for webinar, and for the one-to-one answer, I will send back the PDF of the slide if you are interested. So we're gonna distribute that, and we have recorded this this video for internal training so that we can work on it. So again, thank you all for your participation. And then I'd like to thank Maurice and Shanta for preparing the case study, and Maurice for joining us as an expert to answer the question. You can drop us an email if we missed something or if you want to have more information. Thank you, everybody.
Hi, Pierre.
Yes.
So this is Nicholas here. Can I just add just a few points before you wrap up? So thanks, Pierre, Maurice, and Shanta for sharing. Just a few points which I noticed from your slide, which I'd just like to add some points. These are not questions, just points. I think one is regarding the sinus circle. I think those TACT is actually quite good. I mean, just to add from based on my experience, well, there's other points as well, such as concentration, which Maurice has pointed out. You know, you can have the right chemical, but if you do not have the right amount of concentration, you may not be able to remove, you know, everything, especially the residue inside. The other one is regarding the coverage, right? So just because you wash something doesn't mean that the water goes everywhere. We need to make sure that, you know, the solution actually reaches every nook and cranny, right? So that's why we have things like a coverage test, especially with riboflavin coverage test as well. So that's one regarding the TACT with additional 2C. The other one is regarding the process itself. So of course, you have the pre-rinse, rinse, pre-wash, wash, rinse, final rinse. Regards to the final rinse part, maybe just to add as well, there's besides recirculating final rinse, there's also others like non-recirculating final rinse. Basically, it's like a single pass-through, and basically we have studies. Well, the studies have shown that basically customers can actually can actually have a much lower conductivity in terms of their product if they have, like, for example, like 2 non-recirculated final rinse as compared to 3 recirculated final rinse, right? So basically that actually helps to save water, right? Just think of it as an analogy of you know, going for a bath. If you do soak yourself in a bathtub versus having a shower, you know, a shower is a single pass-through, whereas a bathtub, everything is recirculated, including the residues, so everything gets stuck there. Whereas non-recirculated final rinse is just like a shower, exactly. Everything just goes through, flush down once. Lastly is regarding these washing accessories. So I saw that you have actually shown a variety of washing accessories, which is good. Maybe just to highlight that, and you also did point out in your slide. So in the washer situation, you know, having the standard accessories is good. Well, the main point of the washer is actually of automated parts washer is actually the washer accessory. So in a washer accessory, there's both fixed standard accessories as well as custom accessories. So custom accessories will be able to address the issue of washing complex items. That could even may not be able to, that probably the operator can also wash himself. But there are some parts within some items that even with operator, he or she may not be able to manually wash. So custom washing accessory would be ideal in a sense. And also with a custom washing accessory, basically, if you can actually load up everything into one, you can potentially actually save the number of cycles required to wash the item itself. For example, maybe if you need maybe five maybe cycles using standard accessories to wash items, maybe with maybe one or two cycles can be done using a custom accessory. So yeah, I mean, these are the points, the additional points that I'd like to share based on what I've seen so far. So you know, just some points for you to take note.
I agree with you. And you, there is I put the reference of the different picture we get here, and there is some PE magazine that explains, also ISPs, the PE magazine that explain the difference between a custom washer and a laboratory washer and a dishwasher at home, and what you can do in each of them and things like that. So if you are interested, we could do other seminar in terms of design of equipment or the part of how to watch or CIP and things like that. So valid comment, Nicola. Thank you for that.
Thank you.
So just one logistic, some of the participants actually didn't register, so you won't be included in the email loop. So if you want to receive the material, ask questions, please just drop us an email.
Good. Thank you, Shanshan. Thank you. We're going to continue all day. Thank you very much for joining us.
Thank you.
Take care. Bye-bye.
Bye.
Bye.